All right. So, I'm sorry. What is it? My power tripped off, so my network ran away. All right. So, I was talking about sickle cell disease and how we interpret the permutations. Today, if you are going to marry, the advice is that you need to go for that test to find out whether you are AA or AS or you are SS. Obviously, some SS people from physical appearance you may know, but in science you don't do that. You have to be factual. You go for test. So, but this combination and the way we interpret it is a bit problematic. Now, to give you a simple background as to how a sickle cell disease works, is for simply to understand some little things about genetics. Genetics simply means genes. We are talking about genes. A gene is simply an instruction that tells some part of your body to produce a protein. The entirety of our body is proteins, is meat. So every part of the human body is made of proteins of different kinds. These proteins are being manufactured by something we call the ribosome. The ribosome cannot know that I'm supposed to manufacture a protein for ear or nose or lip or whatsoever. So it has to receive instructions to be able to manufacture that protein. So where does it get that instructions from? Or those instructions from? It gets the instruction from the genes. So the genes tell the ribosome to produce a certain protein. So that is why in the human body we have several genes, about 23,000 genes. So, and the genes tell the ribosome to produce different proteins, just as we have different parts of the human body. Now, the human red blood cell is also a protein, and the manufacture or the design or whatever of the red blood cells is also also done from the ribosome. And some genes would have to also instruct or have to give the instruction to ribosome to produce the red blood cells. Unfortunately, there are this kind of genes that may tell the ribosome to produce a red blood cells that doesn't have a normal shape. The shape will be like a sickle, what we use in harvesting rice or cutting grasses and whatsoever. So when that happens, when that particular gene like that instructs the ribosome to produce that kind of cells, then the person is going to have sickle cells, like I have just described. And when you have sickle cells, then we say that you are suffering from a sickle cell anemia. It means that you are not going to have, anemia simply means lack of red blood cells. So you're going to have a problem with normal blood flow because the shapes of the cells are not normal. So they become very difficult to flow freely in the veins and the arteries of the human body. And those are the reasons why people who are sickle cell, they have problems with so many things. They are prone to, they don't grow as normal as any other person. And then this is purely genetic. It's not something that is caused by any pathogen or is caused by even non-pathogenic. It's not pathogenic, it is non-pathogenic, it is a hereditary disorder. Let me put it that way. So it doesn't come from the fact that somebody you are born with it. Now, but if you are going to marry and then they tell you to go for the test, a genotype test, when you test, the doctor will tell you that, or the lab scientist will tell you whether you are AA, you are AS, or you are SS. Let me say that this AA, A small a, or AS, they mean nothing. They are just symbols to show that you are either having this genotype or you are having genotype. Now, let me say, where do these genotypes come from? They come from the fact that before a human being is born, or is formed in the stomach of the mother, two things come in place. The man combines one part, the woman combines one part. So the parts they combine, this part they combine is not just singleton. Of every single portion of the human body that we see, our eyes or whatsoever are controlled by the genes I've already spoken about. So the genes are not just single entity. Every gene, they are always pair. There are two pairs of alleles that come together, and then that will give you. So one allele comes from the father, one allele comes from the mother, and then it comes to give you the two pair. Now, depending on what, and then these genes I have just spoken about, they are either dominant or they are recessive. The dominant simply means that that particular gene has the propensity to suppress the other one. So it means that if there are two kids and you send them to go and deliver an information, one of them has power, so he will suppress the other one and he will go and deliver the information. So the genes will go to the ribosome to say manufacture this protein, but because one of them is dominant, it will suppress the recessive one and it will go and tell only the information that it has. The one which is the recessive will not reach the ribosome to tell the ribosome that please produce this protein or not. So that's what happened within dominant and recessive genes. Now, and these dominant and recessive genes, they are not only in blood, they are found everywhere. But today, because I want to talk about sickle cell, I'm going to restrict myself to only the sickle cell disease and the permutations. So I'm going to upload this. All right, so if you are watching, I'm going to demonstrate it, and then you see how it is. So you can see I have my plain board here. So let me pull up my pen here. So in every human being, like I've already explained, you now know the genes. So the genes come in pairs like this. So let's say this is human being 1. Let's say this is male, and then human being 2 here, that's a female here. So both of them come together, they also have their individual genes. So what we have here, let's say I have A, that's the AA I spoke about earlier. Then you could also have A and then S. Then you could also have let me clean this one. I think I was supposed to put S here. So the same way, the woman could also have A, okay, A, S, and then also have SS. Now, if we have this allele A, and then you have another allele S, in any case, this is a dominant allele, and then this is a recessive allele. All right, I've just been signaled that the example I'm giving, we need what is, if you are not a scientist, you may not be able to follow. So I'm going to change the SS into. So what I was actually saying was that, so you have AA, you could have A small a, and you could have small a small a. You could also have A small a, and you have small a small a. Now in science, so what I was saying was that this single A, if you have just A like this, that's an allele. The whole of this is a gene. The whole of this is a gene. The whole of this is a gene. These are genes I have circled. These are genes I have circled. But you see that in each of them, there are 2 letters there. Each of them represent an allele. So the allele here is dominant. This is not the spelling of dominant, please just bear with me. The small a here means recessive. Recessive. Now, each time you have a and then the small one here combined, they give you what we call. So when this one and this one are together, the a is more powerful than the small one. So the information that the a is carrying is the only thing you will see in the human being. The one that the small a is carrying, you will not see that one. That's why we call it recessive. So each time, what is it, a male and a female are meeting, they either give out only this or will give us only this. They don't give the two. So one will have to give one out, the other one will have to give the other one out. So depending on whichever one that they give, a particular human being will be formed. So let me clean that portion and show you another thing. So if let's say I have the male here, then I have the female here, and in the course of marriage or in the course of conjugation, let's say I was having a here, a here, it means the two of them are already homozygous. They don't have any problem. If any of them is a small a, it means that they are heterozygous. If they are small a like this, it means they are recessive. The two of them are recessive. Now, the first one, the AA, it means they are free from, let's say, any problem or any recessive character. They are not carriers. The second one means that they are carrying a particular gene which is not expressed, or which that particular gene cannot carry any information because they have a senior brother who is the dominant one. This one, they are recessive dominant, so which means the two the information, because the two of them are small, none can suppress the other, so they can both express themselves. That's what happens. So in genotype testing, when you go to hospital and they test you and they say you are AA, it means you have no problem. If they say you are a small a, it means you are carrying you are a carrier. And if they say you are small a, or you are AS, or you are SS, it means that you are carrying a recessive gene which is expressing. And that is the simple meaning. So now let me show you the combination and where the problem is, where we sometimes get it wrong. So if I go to hospital and do my genotype test, there are ways that they always interpret it. Are you AA, or you are AS, or you are SS? So let's assume that the man is here and the woman is here. So let's say the man is A, married to a woman who is A. So it means the two of them have tested and they are AA. They have no problem. They are 100%. If one of them is A, small a, and the other one is, let's say, A, there's no problem. That's 100%. And it's vice versa. Whichever way you can look at it is vice versa. It's either you go this way or you come this way. It's the same. Now, if let's say the man is A, small a, and the woman is also A, small a, this is not a good combination. It's not a good combination at all, because there is a 25% chance that one of the children will be sickle cell. So I'm replacing the small a with S. It's the same thing. Wherever you see the small a, it means S, which means sickle cell. So if one of them is like this and the other one is small a, all the children are going to be normal children. So it means if one parent is completely dominant and the other one is a sickle cell patient, when they give birth, none of the children will be sickle cell. They are all going to be, but all the kids are going to be carriers. They are going to be carriers of sickle cell, which means their great-grandchildren are likely to be sickle cell. That's how it means. Now, if you are, let's say, A and then small a, and then somebody is, and then the other person is sickle cell. If you are a carrier of the sickle cell gene and the other one is completely sickle cell, it's not a good combination, because 75% of the kids are going to be sickle cell. And of course, this is a no-go area. If the combination is like this, it's a no-go area. It's a no-go area. It's 100% everybody there is going to be sickle cell. Now, but there are some of the combinations here that are good. Others are there, they are not good at all, and don't try it. Now, the first one here is a good combination by all means, which means both man and woman, if they are homozygous, it means they have AAAA, they have no problem. The second combination that is good is this one. If one of them is a carrier and the other one is completely dominant, it is a good combination. There's no problem about that. Now, the third one is if one of them is completely dominant and the other one is completely recessive. When they marry, none of the kids will be sickle cell. None of them will be sickle cell. So it's also like 100%, but the problem is that all the kids are going to be carriers. They are going to be carrying the sickle cell gene in them. All right, so which are the combinations that are not too good? The very bad one is this one. I think I should change my let me erase that one, and then I take a different pen and circle it so that we can see it. Let me take yellow. This is a very bad one. Following close to that bad one, 100% bad, is another. This is 75% bad, and then this is another one that is also bad. Now, in all of this, it is easy for people to interpret the all those in yellow, but there's one that, so all those that are in yellow, the AA and AA, people will definitely not go for that. You won't find a sickle cell patient going to marry another sickle cell patient, no. And you won't find someone who is a carrier who wants to marry a sickle cell patient. Now, the only place where you find that people try to compromise is when the two of them, the man and the woman, are both carriers. So one of them, none of them is sickle cell, but each of them is carrying the sickle cell gene in them. So I'm going to demonstrate that, and then you will see where the misconception is always coming in. Let me erase everything, and then I will then pick only that portion. So if the man is A, small a, the woman is A, small a. Now, in all the textbooks I have seen, and we've been studying this thing, this is how scientists always demonstrate. They will say that, okay, so if they marry, and the man happen to give this allele, the woman give this allele, the child is going to be A. If the man donate allele A and the woman donates a small allele A, then the child is going to be A small a. If in the other instance the man donates the small allele A and the woman donates this one, the capital one, then it's going to be capital A small a. And the last combination, when the woman donates small a and then the man also donates small a, the child is going to be small a. So in in genotype combination, wherever you see small letters, it means the person is a recessive dominant. So if these particular alleles were coding for sickle cell protein, it means this particular child will be SS. Now this particular child here is going to be a carrier hetero. This child here is going to be a carrier hetero, and then this child here is going to be a normal child. I hope you are getting you are following the point now. Now, if you look at the way I have done it, and this is the way you see it in textbooks, the misconception and the interpretation is that this is the first child, this is the second child, this is the third child, and this is the fourth child. This was what informed me to do this video. Somebody did this analysis and said, okay, in that case, if I marry, I'll give birth to 3. I will not give birth more than 3. So if I give birth to 3, the possibility is that I will have all of them to be normal children. So it means the fourth child, who is likely to be SS, I will not give birth. So that one will not come. This is a very wrong interpretation of data. I don't know the one who provided that advice, but this kind of thing is always theoretical, that scientists try to explain to let people understand how the combinations are done. Let's assume that the woman has given birth to about 8 or 9. My mother gave birth to 9 of us. How is this going to be done? The only way to interpret this data is that it means that if a career man marry a career woman, there is a 25% chance that all the children they are going to give birth will be sickle cell. There is a 25% chance that all the children they are going to born will be normal children. There is a 50% chance that all the kids they are going to give birth, they will be carriers. Carriers are practically like normal children, but which means they are also carrying the gene. And this is an example. That's what I want us to know. So it does not mean that when we see this kind of combination in our textbooks, it doesn't mean that the first kid is naturally going to be a normal one, followed by something. No, it's not in that order. This is just a demonstration. So it could have been that if you can see my screen here, if we were having, let's say, if the combination, let's say another one here, it could have been that all the combinations you will see, like it could have been SS. The next time that they are going to born, the man will still give another S, the woman will still give another S. The next child they are going to born, they will still give SS. The next child they are going to born, they will still give SS, in that order. And then you find that all the kids in the house will be SS, they will all be sickle cell. There's that chance. There's also that chance that all the kids could be normal. And there's also the chance that all the kids could be heterozygous or carriers. That's the point I was trying to make. So in interpreting this data, please, let's be careful. And I think that with everything, put everything together, if you are a carrier and then your woman too is also a carrier, please, if you want to compromise, you can compromise, but you should know that you can give birth to 10 and all the 10 kids will be sickle cell. All of them will be sickle cell. They will have sickle cell anemia. They will suffer from sickle cell. So that's the misconception I wanted to clear. And in a case you do not get anything clear, please, I am still here to answer any question. So this is what it means. If you now have, if the child then inherit, I think I need a different pen now. If the child inherits A from the father, and that child is called a carrier. If the child inherits A, that child is called a normal child. If the child inherits S from one parent, S from the other one, that child is called a sickle cell or a sickle cell patient. Why? Because I've already said that this is a gene, and this gene will carry an information to some cylindrical object called ribosome. But because this A is dominant and then this S is not dominant, the information they are carrying to this ribosome that is going to produce some protein, the one that is carried by the small s will be suppressed. It will not reach here. So it's only the A that will come here to deliver the information. The S will remain outside here. It will not be there. So the information that the ribosome will receive to produce the protein, it will only be the information carried out by the A, the allele A, because it is dominant allele. The S is recessive. Now, when it comes to this one, the normal, the normal combination, it means that the child has inherited a dominant allele from both parents. So, in the ribosome of that particular child, all this particular gene, all of them would always come here to deliver the information. They will all come here. A will be there, another A will be there. So the whatever information that A is carrying, the other one is carrying, it will be delivered here. So because both of them are dominant, so one cannot suppress the other. If it is also SS, both of them are weak. One is not. Both of them are weak, so they can all move at the same pace. So they will all also come to the ribosome and deliver the same information. And so if both of them come here and deliver the same information, the information that is available to this ribosome on this particular one will be that produce a blood called HBS. And this particular blood is what gives us a blood with this particular shape called the sickle cell. But these two we have here, the one we have here and then this particular place, they are going to produce, they are going to carry an information that will tell the ribosome to produce a blood that will have a normal shape. It will be like this. The shape will be, let's say, this way. It's not going to be like this sickle something. So if it is a normal cell like this, with a normal shape like this, it can flow freely in the veins and arteries of the human being. Because this one has this kind of shape, their membranes have overgrown, so nutrients leak. And when the nutrients are leaking, that's what makes sickle cell patients lack. They always lack this red blood cells. It leaks most of the nutrients. And it's also the reason why they are malaria advantage. But that's a different video I'm going to do on a different day. So, in a nutshell, this is how sickle cell analysis is being done. And this is how we should interpret the data. Understand your partner, and you should know the risk that you are taking before you enter into it. So that's all that I wanted to explain today as far as sickle cell permutations and interpretation is concerned. Thanks if you were watching, and God bless you. If you have any question that you have asked, I will try and look at it in the comment section, and then I will answer it accordingly. The next video I'm going to do, possibly maybe tomorrow, will be how we interpret high blood pressure. The systolic reading and the diastolic readings. You see, high blood pressure is, we are looking at how the heart receives blood, adds oxygen to it, become oxygenated, then distribute it to the body. So there's an inflow and there's an outflow. So when the heart is pumping, the muscle is pumping, it receives and it pushes out. So we have systolic pressure and then the diastolic pressure. So which one? So how do we interpret it? If the systolic is this number and the diastolic is this number, how do we interpret it? What I've noticed is sometimes you just go to hospital and then, or not hospital, maybe you just go to someone and the person will just check. Especially these days, you see in the market, people are carrying the blood pressure machine to test people. They just pump there and say your blood pressure is high. They have not even considered the temperature of the day, the kind of work you are doing, your gender, your blood group, and there are a lot of factors you need to put together before you can interpret somebody's blood pressure and then say that the person is either having a high blood pressure or it is normal or it is low. And if they are normal, if it is low, what do they mean? At what point do we say the blood pressure is actually not normal? Thanks very much for watching, and I am so grateful. I'll meet you tomorrow with another edition. My name is Bigba Bright, and I have passion for always breaking scientific issues to a very layman's understanding. See you again.